Hello friends, good afternoon and welcome to the EduSite Network. Our topic of discussion today is part of our ongoing series on immunology, and this shall be the fourth lecture as part of it. The specific topic, friends, for today is antibodies, structure and function generation of monoclonal antibodies, and for this very lecture we have with us in our studio our same subject expert who has been associated with us all throughout the series, Dr. Hardeep Kaur, Assistant Professor, Department of Zoology, Ramjas College, University of Delhi. With this brief introduction, I would like to welcome ma'am to our studio. Thank you so much, ma'am.
Thank you, Urvashi. Friends, so today's our topic is antibodies, and you all know it's a very important part of our immune system. Today we will be dealing with the structure and the function of various types of antibodies, and also what is a monoclonal antibody and what is the method of generation of monoclonal antibodies. Now, coming first to the definition of antibody, what exactly is an antibody? An antibody, also known as immunoglobulin, is a large Y-shaped protein produced by plasma cells. They are recruited by the immune system to identify and neutralize pathogens such as bacteria and viruses. The antibody recognizes a unique molecule of the harmful agent called an antigen, or more precisely the specific epitope of an antigen. In our last lecture, I specifically mentioned what are antigens and what are epitopes. As I told in the last lecture, antigen could be a big molecule. So, any particulate or a big antigen, be it a microbe, be it a parasite, whatever it is, it can have multiple small units which are called epitopes, and against each of the epitope which is also known as an antigenic determinant, you can have a specific antibody to it. So, for a particular antigen, there could be numerous epitopes, and for each epitope you have a specific antibody which reacts to that epitope. Now, with this definition, let us further understand what is an antibody, where is it located. Antibodies, as we all know, are generally produced by B lymphocytes. They could be present as the membrane receptor of the B lymphocyte, or it could be in the secreted form in the blood plasma. Now, it shares structural features, binds to the antigen and participates in number of effector functions. They collectively are known as immunoglobulins. I will come to the point where I will explain why they are called immunoglobulins. They do not kill anything. Let us understand, they are the effector molecules. They just provide the kiss of death. So, they do not themselves kill anything. Now, they belong to a superfamily called immunoglobulin superfamily. Now, there are numerous molecules like T cell receptors, the class 1 and 2 MHC molecules, and of course antibodies which have similar domain structures. Most of these are proteinaceous in nature, and because of the similarity of structure, we call them to be belonging to the same superfamily, that is the immunoglobulin superfamily. However, unlike antibodies which can bind to the antigen, most of the other molecules directly may not be binding an antigen, but because the fact that they have the same homologous structural affinity, they are hence put into a superfamily called the immunoglobulin superfamily. Now, coming to the history of immunoglobulins or antibodies, Tiselius and Cabot in 1939, they isolated immunoglobulins and found them to be in the gamma globin fraction of the serum. Hence, the name globulins came into being, and because it is involved in the immune system, we started calling them as immunoglobulins. Rodney Potter and Gerald Edelman, they were the ones who deciphered the structure of IgG, a particular type of immunoglobulin, and for this they received a Nobel Prize in the year 1972. As you can see, we have Potter on the upper of the slide, and this is followed by Edelman. They were the real people who actually founded the structure of immunoglobulins. Now, these all molecules, as I was just telling you, they belong to the immunoglobulin superfamily. Here you can see we have an IgM, which is a type of immunoglobulin. We have a T cell receptor, we have MHC molecules, as well as the immunoglobulins which are specifically located on the membrane of B lymphocytes. All of them, if you see the structure of them, they have the same domain molecules, they have the same similar structure. hence belonging to the immunoglobulin superfamily. Now, if you come to the structure of an antibody which was deciphered by Porter, antibodies are heterodimers. They are made up of 2 light chains which are each of 25 kilodaltons and 2 heavy chains which are 50 kilodaltons in each. If you go to the next slide here, you will see that this is a Y-shaped structure of an antibody, wherein the central 2 chains are the heavy chains, the outer 2 chains are called the light chains, and they have different regions to it. For example, the heavy chain has VH region, CH1 region, CH2 region, CH3 region, while the light chains have the VL region and the CL region. Now, coming back, so each light chain is bound to the heavy chain by disulfide bonds. Again, if you see in this slide, the light chain is connected to the heavy chain by a disulfide linkage. Now, this particular linkage is very important. It provides a binding to both the light and the heavy chain. It is a covalent binding. Besides this covalent disulfide linkages, there are also certain non-covalent bonds also present or bridges also present. It could be in the form of a hydrogen bond, ionic bond, or certain other interactions which also provide help for these 2 chains to interact with each other. Now, coming back to the first slide again, each heavy chain is itself bound to another heavy chain by disulfide linkage. Similar goes for the light chain. Each light chain is bound to the other light chain by the disulfide linkages again. Now, first 110 amino acids of the amino terminal vary of both H and the light chain and are called variable. Now, as you can see in this slide, the VH and the VL region are the variable regions of the heavy chain and the light chain respectively of the antibody molecule. Now, again they are hence called VL and VH, and if you see again in this structure, you have the CH1, CH2, CH3 regions of the heavy chain and the CL region of the light chain, which are called the constant regions of, because their sequence remains constant. They do not have much variability, hence called the constant regions of the heavy chain and the light chain respectively. Now, again the antibody possess a CDR, that is the complementarity determining region. This is the region where you have the variation, specifically the variable region where binding of antigen to the antibody molecules takes place. Remaining regions, as I just told you, the constant region, are very similar within the same class of an antibody. Now, again coming back to the structure, there are intra-chain disulfide bonds, heavy chains have VH1 and either CH1, CH2, CH3 along with CH4 in certain antibodies. The light chain has VL and CL region. There is a very important hinge region, as you can see, as it is marked as hinge. This is the region which is very much rich in purine residues and is also the site of digestion, enzymatic digestion of the antibodies. This hinge is found in IgG, IgA and IgT type of antibodies or immunoglobulins, but are absent in IgM and IgE. It is also rich in cysteine residues which are responsible for the disulfide bridge formation. Now, initially, to find out the exact structure of the antibodies, various important experiments were performed. In some of the experiments, as to how these chains are bound to each other, many of these experiments were performed, and Porter for the first time found out after digestion with papain that 3 fragments are obtained. As you can see, if you go for the papain digestion of an antibody molecule, it leads to formation of 3 fragments, 2 Fab and 1 Fc region. Now, this papain digestion will cause these Fab fragments to form, which has an ability to, they are monovalent fragments and hence they individually can bind to an antigen. Fab, as the name suggests, it is the region where antigen binding take place, while the Fc fragment which is formed is the fragment that undergoes crystallization, hence called Fc fragment. Now again, in another experiment done by Nizonov in 1950s by pepsin digestion, which is another proteolytic enzyme, it was found that Fab2 fragment was formed, which shows antigen binding and could lead to precipitation of an antigen. However, the recovery of Fc was not possible because it completely got digested. As you can see in this slide, if you have a pepsin digestion of an antibody molecule, you get a Fab2 fragment which has the ability to bind an antigen, and because it can bind an antigen, it is retaining both the antigen binding sites, it can cause precipitation of an antigen as well. However, the Fc fragment has undergone degeneration. In another experiments using mercaptoethanol reduction, the disulfide bonds were reduced and the separate H chains and L chains, that means heavy chains and the light chains, were obtained. So, all these experiments helped the scientists to find out the real structure of an antibody molecule. Now, coming to the facts about immunoglobulins. Immunoglobulins, as you can repeatedly call them as antibodies also, they can be in a secreted form or in a membrane bound form. The membrane bound differs in the carboxy terminal end. It has an extra spacer of 26 amino acids and a hydrophobic transmembrane sequence. Now, as you can see in the next slide, the one which is bound to the membrane is having an extra 26 amino acid long spacer molecule, and it is having a cytoplasmic tail as well. And on the other hand, the one which is in a binding form to an antigen, it is devoid of this spacer region, and it does not have any cytoplasmic chain also. Now, in addition to binding to an antigen, the antibodies can have various other functions. It can promote phagocytosis. For example, it can bind to the or opsonize. Opsonization is a process by which an antigen can be made compatible, or the antigen is made more palatable to the immune system. The antibody coats the antigen in such a manner which is called opsonization, and hence present this antigen to the immune system. This process is called opsonization. Specifically, in this case, it is the phagocytes which are being triggered by opsonization of an antigen using antibody molecules. Now, the second function which is very important is to activate the complement. Complement are a group of proteins which are very important. They form a very important part of immune system, which you will come to know in another lecture. This complement has a tremendous role in killing many antigens. Now, antibodies specifically on the constant region of the heavy chain have certain areas where the complement proteins get bound, and then the complement system, which is a cascade mechanism, get activated. So, antibodies have a tremendous role in activating the complement system, which is important in killing the antigen. Besides that, the antibody also have a role to perform in ADCC, which is antibody dependent cell mediated cytotoxicity. In this case, the antigen which is presented by certain cells, the APCs etc., is bound to an antibody, and antibody is forming a sandwich between the killer cell. The killer cell could be a T-type cytotoxic cell and an antigen. So, this process is dependent upon antibody. Antibody is between it is like a sandwich, you have two breads and slices and you have something in between. This something in between filler is what antibody is doing in this process. Now, some antibodies can also cross the epithelial layers to be excreted through mucus, or can cross placenta also. This is another very important property of an antibodies, that they are capable of crossing the epithelial layers as well. Now, this I had already explained. Now, coming to antibody antigen interaction, a separate lecture can be given on this antibody antigen interaction. However, I will just touch briefly upon it, that smaller antigens, because I had already told in my previous lecture, the antigen binding to the antigen binding site of an antibody can bind in specific manners. For example, the smaller antigens can just fit into the pocket on the variable region of the antibodies. However, the larger globular antigen, they interact with the flatter region of the variable region of an antibody. This we had covered in our last lecture also. Now, coming to the classes of various immunoglobulins. Now, sequencing of several immunoglobulins have found that there are 100 to 110 amino terminus hypervariable regions or complementarity determining region or the CDR region. This is the region where the antibody binds to an antigen. Because there could be many different types of antigens binding to the antibody, this region is highly variable. The sequence present in this region are extremely variable, and hence it is catering to numerous types of antigens that a immune system can come and combat. There are basically 5 basic sequence patterns. Now, these 5 basic sequence patterns are dependent upon the constant region of the heavy chain. The constant region of the heavy chain can have 5 epitopes or they can be of 5 different types. It could be alpha, it could be gamma, it could be delta, it could be epsilon or mu. Depending on it, the antibodies are or the immunoglobulins are called as IgA, IgG, IgD, IgE and IgM. The above classes, as I already told you, are also called isotypes. Each class further can have a kappa or a delta light chain. Now, I would like to bring back your attention to the structure of antibodies. If you see the variable region in the heavy chain, VH region, CH1 region, CH2 region and CH3 region. Now, the CH1, CH2 and CH3 are of 5 different types, as I just told you, alpha, delta, mu, gamma, epsilon. Depending on that, we give a type of an immunoglobulin, whether it is IgM, IgD, etc. Now, similarly, if you see the Cl region of the light chain, you will see that the Cl region of the light chain can also be in 2 forms, either in the kappa form or the lambda form. But for a particular immunoglobulin, it has to be both. The light chains has to have a kappa constant region or the lambda's constant region. It cannot be that an immunoglobulin can have one kappa region in one chain and another lambda region in another chain. Both the chains got to have either kappa or lambda. In humans, it has been found that the light chains are generally having 60% of the light chains in immunoglobulins have the kappa chain, while the other 40% had lambda chain, while in mice it has been found that 95% of them have kappa and only 5% of them have lambda. There are minor differences which further lead to the subclasses for IgA and IgG immunoglobulins. The IgA is further being described as IgA1 and IgA2, while IgG has further been called as IgG1, IgG2, IG3 and IG4. Now, coming to the structure of various types of immunoglobulins, as you can see, IgG is generally monomeric in nature. We will come one by one to each one of them. These are the various or the 5 different types of immunoglobulins, where we have IgD and IgG, IgE. IgA is generally present as a dimer with a J chain which attaches to chains of IgA antibodies or 2 molecules of IgA, while IgM is generally seen as a pentamer. Now, coming individually to each of the subtypes, the IgM or the immunoglobulin M. Now, immunoglobulin M is actually the primary response antibody. See, whenever there is an antigen which is coming into and the immune system is coming in contact with an antigen, there are 2 types of responses. One is called the primary response and one is called the secondary response. The primary response is not very enhanced. However, coming again in contact with the same antigen second time, the body or the immune system produces tremendous amount of secondary antibodies. The primary response main antibody is immunoglobulin M. It is present as 5 to 10% in the serum immunoglobulins. The total percentage in the, if you consider it in serum, the total percentage of immunoglobulin M is only 5 to 10%. It is generally monomeric if it is expressed on the B cells. However, in a secreted form it is pentameric in nature. Now, it activates complement and it is very good at agglutination, not as good as precipitation, however, it is very good in agglutination. There is a J chain which allows IgM to get transported across the epithelium layer also. The molecular weight, because it is a pentameric in nature, the molecular weight, as I just told you, the molecular weight of the heavy chain is 50,000, the light chain is 25,000. So, each molecule is roughly having 1,50,000, or because it can be variable also, and in this case because they are pentameric in nature, so the molecular weight of IgM happens to be 9,70,000 Daltons, while it has a sedimentation coefficient which is also very high, which is 19S. So, we must remember IgM is a pentameric antibody. It is produced in the primary response against an antigen, it activates complement and it is present almost at a concentration of 5 to 10% of the serum immunoglobulins. Coming to IgA, it is almost 10 to 15% of the total serum immunoglobulins. It is a predominant secretory antibody. Now, if you remember, once a child is birth takes place, after parturition, for the first 3 days the milk which is produced is called colostrum. This colostrum is extremely rich in IgA antibody. In fact, for the first 3 months that the milk is produced, it provides effective immunity to the growing baby. So, this milk is very important, and that is the reason why it has been promoted that breastfeeding is very important, because this milk is predominantly having IgA antibody which provides immunity to the newborn. Now, it is a predominant secretory antibody. As you can see in this slide, the GA chain and the secretory component is the one which helps it in transportation across the intestinal wall. So, it is one antibody which does not get digested by the enzymes present in the digestive system. The J chain makes the IgA more resistant to acids and enzymes that are found in digestive tract. It can cross-link large antigens. Mostly, as I just told you, in the secretory form it is existing as a dimer. The molecular weight being a dimer is 3,85,000 and the sedimentation coefficient is about 11S. Now, in the slide you can see the diagram of IgA. As you can see, it is a dimer and this J chain and the secretory component. In the next slide you will see there is a secretory component present. This secretory component along with the J chain gives a protective protection to the IgA, the secretory IgA, through all the intestinal or the digestive enzymes present in the digestive system. Now, it is, as I just told you, it is mostly present in the seromucous secretions, for example, the breast milk, saliva, tears, the mucus of the bronchial, genitourinary and digestive tracts. Coming to IgD, now IgD is generally found bound to the B cells. Now, majority of the function of IgD antibody is not known, but people think that probably it functions in the activation of certain B cells. It is only about 0.2% of total serum immunoglobulin. However, together with IgM, it is considered to be the major membrane bound Ig on the mature B cells. Of course, it is monomeric in nature. And coming further to immunoglobulin G, a very important immunoglobulin type. This is the most abundant. Almost 80% of our total serum immunoglobulins are composed of immunoglobulin G. It is monomeric in nature, but it has 4 subclasses in human. This is one immunoglobulin that can cross placenta also. It is again involved in complement activation. the IgG1, G2, G3, G4, these are the subtypes of immunoglobulin G. As you can see, the structure of Ig3, the molecular weight of Ig3 is the largest because of the bigger heavy chain constant region. The concentration at which these subtypes of immunoglobulin G are present is almost 66% of IgG1, 23% of IgG2, 7% of IgG3 and the rest IgG4 4%. Now again, as you can see in the diagram, it is having a monomeric structure. So the molecular weight has to be roughly around 1,50,000, but the actual molecular weight of an IgG and an average we consider it as to be 1,46,000 molecular weight. It has a sedimentation coefficient of 7s. It is a very important immunoglobulin in the secondary response. As I just told you, once an antigen comes and the immune system gets ready to combat, this antigen, if it comes second time, in the primary response the predominant antibody is IgM, but the same antigen, if it comes into and tinkers our immune system to then or pokes our immune system, then the secondary response which is produced is predominantly having IgG antibody. So it is a secondary response antibody, and as I just told you, almost our immunoglobulin pool has 80% of immunoglobulin G. Coming next to immunoglobulin E, we all know this immunoglobulin is specifically involved in allergic reaction. It is also involved in parasitic infections. The concentration in the serum is very low, but it all depends. If there is an allergen present, the concentration of IgE can actually go high. It is responsible for the symptoms of hay fever, asthma, hives and anaphylactic shock. This is the extreme case of allergy where the person can actually die because due to any allergen present. Now, how exactly this immunoglobulin E? It is mostly present in the surface of the basophils and the mast cell. Repeated antigenic or an allergen explosion activates IgE. The binding of allergen to IgE activates the mast cells and basophils, and they undergo degranulation and release of histamine. This histamine is a substance which is leading to an allergic reaction, and hence depending upon the severity of the allergic reaction, it could be a hives, it could be a hay fever, or in an extreme case it could be an anaphylactic shock also. Now coming to the variations in immunoglobulins, the immunoglobulins can have variations which could be called isotypic variations, allotypic variations or idiotypic variations. Now in case of isotypic variation, there is a difference in the constant region from one species to another. Any individual, any healthy member of a species will have genes for gamma 1, gamma 2, gamma 3, gamma 4 or alpha, mu, epsilon, delta. These are the isotypes as I already described that these are the isotypes which are coming from the constant region of an immunoglobulin. Any healthy individual of a species will have all the isotopes. However, in case of allotypic variation, there is a difference at an allelic level, at a particular locus. Not all individuals may have a particular allele. For example, in case of IgM, IgG3, G3M is an allotype which has phenylalanine at position 436. This particular allotype may not be present in all individuals. So, allotypic variation can vary. Not every individual is going to have an allotypic variation. The last type of variation is called an idiotypic variation, where there are differences in the variable region. The variable region, we all know, the VL region or the VH region of light chain and heavy chain respectively have high variation. They have hypervariable region. This is called idiotypic differences, which will differ even on antibodies of the same isotype, because the variation is the one which is causing the antigen of various types to bind with it. So, idiotypic variations are going to be there in all individuals. Now, coming to the discussion of polyclonal antibodies versus monoclonal antibodies. You must understand that whenever an antigen comes and the immune system combats an antigen, as described in my last lecture, an antigen could be a big molecule with many epitopes or antigenic determinant sites. Each epitope is capable of binding to a particular antibody. So, rather I would say each epitope can lead to an antibody production, which could be a monoclonal antibody for a particular B lymphocyte. So many different types of B lymphocytes will be triggered by a particular antigen. One antigen leading to various types of antibodies. So, at any given time, the serum we have is a polyclonal sera. It has a mixture of various types of antibodies because we have numerous antigens coming into our body, each antigen having numerous millions of epitopes in it, hence triggering millions of antibodies. So, at any given time we have a polyclonal sera. It is in fact very important because polyclonal sera is the one which is causing the immune system to take care of various different types of antigens. So, polyclonal antibodies are those antibody preparations from immunized animals. They consist of complex mixture of different antibodies produced by different B cell clones. However, whenever we have certain therapeutic uses or diagnostic uses or to be used in the research, these polyclonal antibodies are not much of a use. We require monoclonal antibodies to perform our experiments. Now, monoclonal antibody is a homogeneous antibody preparation which are produced in the laboratory and they consist of a single type of antigen binding site and generally produced by a single B cell clone. Now, as you can see, polyclonal antibodies can form lattices with homogeneous monomeric protein antigen because each antigen can interact with a different epitope of an antigen. Now, on the other hand, monoclonal antibodies do not form lattices with homogeneous monomeric proteins because only they can bind to only one particular type of an epitope on an antigen. Now, coming to the history of monoclonal antibodies, in 1975 the Hybridoma technology was deciphered by George Kohler and Cesar Milstein, who devised a method to obtain monoclonal antibody by Hybridoma technology. For this they received a Nobel Prize in 1984. In 1988, the Nobel Prize in Medicine went to Greg Winter, who developed the technique to humanize the monoclonal antibodies. Continuing with the history of monoclonal antibodies, in 1986, first monoclonal antibody Muromonab CD3 reached the market. In 2003, the first fully human monoclonal antibody adalimumab came into the market. Now, this is a list of the FDA approved monoclonal antibodies which are specifically used for the cancer treatment. As you can see, the name of the drug is alemtuzumab, which is Campath, and the type of cancer which is cured is in this case chronic lymphocytic leukemia, Avastin, bevacizumab, breast cancer, colon cancer, lung cancer, glioblastoma multiforme and so on. This has the source for this slide is from the Mayo Clinic. The rituximab, which is called Rituzan, is used against non-Hodgkin's lymphoma and various other drugs. This is a list of the FDA approved monoclonal antibodies specifically for cancer treatment. Monoclonal antibodies have other uses also against other diseases also, but this slide is specifically for the cancer treatment. The monoclonal antibodies also have other uses in therapeutic as well, not only in therapeutic but also in diagnostic, which we will cover later. Now, there are two ways of, as you can see in this, there are various ways of getting monoclonal antibodies. I will come to it. This particular slide shows an antigen with different types of epitopes. If this antigen is injected into a mice and their serum is isolated, naturally because there are 4 different types of epitope, this is hypothetical statement where we are using only 4 different epitopes just to make the things easier to understand. Now, we will have antibody against the one epitope which is called AB1, AB2, AB3, AB4. Now, if we directly isolate the serum from this rat, this would be a polyclonal serum. Now, if we isolate the spleen and make sure that there are plasma cells and these plasma cells are further bound to the myeloma cells to give it immortality and undergoes hybridization to get hybridomas, and then further selection is done, we will be able to get monoclonal antibodies separately, AB1, AB2, AB3, AB4. This looks very easy, but it is not a very simple technique. We have a hybridoma technology. Now, before I come to the hat selection or the hybridoma technology, I would like to say that there is another naturally occurring monoclonal antibodies in nature. These monoclonal antibodies are specifically from the myeloma proteins from the plasma cell tumors. It is a cancer of antibody producing cells, for example, in case of multiple myelomas. The neoplastic transformation of single plasma cells can lead to a tumor which will be secreting a homogeneous monoclonal antibody. Now, this homogeneous monoclonal antibody can actually go and the level can increase up to 95%, and hence the serum can be isolated, and 95% of the immunoglobulin present in that serum would be the homogeneous monoclonal antibody, because it is a tumor of a plasma cell. Now, these tumors have not only been isolated in case of humans, but also in rat, mice, horse and dog. They can be induced into two strains of mice also, the bulby C and NZB, where by injecting mineral oil into the mice, into the peritoneal cavity, we can induce the myeloma cell tumors. Now, these tumors have a very important, these have not only have an immortality characteristic of immortality because they are tumorous in nature, but they can be maintained in various rat or mice species, and you can actually have banks of these myeloma tumors. That is a natural way of getting monoclonal antibodies. There is another method, as I was just describing, the hybridoma technology. In hybridoma technology, from the spleen of a recently immunized mice, you can obtain an antibody producing lymphocyte. This antibody producing lymphocyte is then hybridized to the myeloma cell, which this particular cell line must have two characteristics, that it is lacking in the antibody production and it is lacking in HGPRT enzyme. HGPRT is hypoxanthine guanine phosphoribosyl transferase enzyme, a very important enzyme which utilizes hypoxanthine and guanine, which along with 5-phosphoribosyl phosphate leads to the formation of inosine monophosphate and guanosine monophosphate, very important nucleotides. Now, this HGPRT is very important to utilize hypoxanthine and guanine and convert it into nucleotides IMP and GMP. Now, the myeloma cell line is lacking in the antibody production, it is also lacking in HGPRT. However, the normal spleen cell producing antibody from the mouse which has been immunized by a particular antigen will be producing a particular antibody. Now, when it is hybridized to the myeloma cell in a medium called the hat medium, the hat selection is used to select for growth of hybrids and against the growth of the parental myeloma. Now, this hat medium is actually a combination of hypoxanthine, aminoterrin and thymidine. Now, hypoxanthine and thymidine are the raw materials which are required for the HGPRT enzyme. However, the aminoterrin, which is a folate antagonist, it blocks. Now, any cell which will utilize two pathways, one is called the de novo pathway, the other is called the salvage pathway. Now, aminoterrin, which is a folate antagonist, does not let the cell to follow the de novo pathway. So, the only option for any cell, whether it is a myeloma cell or a spleen cell, is to follow the salvage pathway. Now, HGPRT is a very important enzyme of the salvage pathway. Now, the normal spleen cell or the lymphocyte, because it is having an HGPRT, can produce the nucleotide IMP or GMP using the salvage pathway. However, on the other hand, the myeloma cell line could not produce these nucleotides through the salvage pathway because it is HGPRT negative. Now, when both of these are fused using polyethylene glycol, which is called a fusion agent, as you can see in this slide, you will have 3 types of cells which will be produced in the culture. One would be the normal lymphocytes, one would be the myeloma cells, and there would be another category of cells which are the hybrid cells or the heterokaryon. Heterokaryon is when the 2 nuclei coming from diverse cells, they fuse to each other, they form heterokaryon, and hence the hybrid cells are obtained. Now, hybrid cells between the lymphocyte, the normal lymphocyte from the spleen of the mice and the myeloma cell will be produced. Now, in this hat medium, the normal myeloma cell, the myeloma cell of the parent myeloma cell will not be able to survive because it does not have HGPRT. So, in any case, it cannot go through the de novo pathway, it cannot go through the salvage pathway. So, a normal myeloma parent will not be able to survive. The normal lymphocytes, they perish after 1 or 2 division. So, the only cells which can survive the hat medium is the hybrid cells, and because it is a hybrid cells, it is getting HGPRT from the parent spleen cell or lymphocyte, and hence it can use the salvage pathway. It is also getting the property of producing a particular antibody, and now because it is getting the immortality from the myeloma parent, hence it is an immortal cell line of this hybrid cell, could be called an immortal cell line, producing a monoclonal antibody or one homogeneous type of an antibody of our choice against that antigen with which we had infected the mice in the previous in the parent cell. So, it is producing a monoclonal antibody of our choice. This technology has been extremely useful in getting monoclonal antibodies. However, the usage of this technology is very limited in case of humans because one, to get a myeloma cell line which is HGPRT negative, not producing antibodies, has the property of immortality, can be used in the hybridoma technology. It is very rare to get such a myeloma cell, but nevertheless it is a very important technology to produce a monoclonal antibody. Coming back to the slide, here as you can see, the spleen cell, the normal parent is bound to the myeloma cell, these two are the parents, myeloma cell is lacking HGPRT. As you saw, you have a fusion of the parents leading to production of hybrids. When these are transferred to the hat medium, the parents normal lymphocyte after 1 or 2 divisions will perish. The myeloma cell will not survive because it is HGPRT negative. The only cells that survive is the hybrid cells, as shown, it is yellow in color. This is the hybrid cell which is formed between which is a hybrid between the parent lymphocyte and the myeloma cell. It is producing the antibody, monoclonal antibody which we needed, and it is capable of immortality. That means it can be grown and further checked, and this is an extremely important technique. Now, hybridoma selection, which we call it as hat trick, is employing actually 3 important features. The myeloma cells have been genetically engineered such that they cannot use hypoxanthine and thymidine in the hat medium as source for nucleic acid biosynthesis and hence will die. The spleen cell, as I just told you, will have a limited lifespan, will die after 1 or 2 divisions. Only B cells that are fused with the engineered myeloma cells will be able to survive in the culture when further grown in a hat medium. Now, what are the advantages of the monoclonal antibodies? There are numerous advantages of the monoclonal antibody. They are consistent, they are limitless supply of a specific reagent, and they are more easily tested for cross reactivity. Further application of monoclonal antibodies, if we see that monoclonal antibodies can further be used for diagnostic tests, for detecting pregnancies, numerous pathogenic microorganisms, measuring blood levels of various drugs, matching histocompatibility antigens, detecting antigens which are shed by certain tumors. So, hence they have a very good role in diagnostics tests. The antibodies are capable to detect even the tiny amounts in picogram per ml of molecules. For example, most of the kits which are for the detection of pregnancy utilize monoclonal antibodies. They also help in diagnostic imaging. For example, monoclonal antibodies that recognize tumor antigens are radiolabeled with iodine I131. They have a very important role as immunotoxins. Now, I want to elaborate in this. The monoclonal antibodies, if they are tagged with certain toxins like ricin, shigella, we know these are toxins. Now, these monoclonal antibodies, if they are specific to a particular tumor, these will go inside the body and will go and bind to those tumor cells. Now, if they are attached to a ricin or a shigella toxin, the toxin will finally kill those tumor cells. So, hence monoclonal antibodies has important role as immunotoxins also. Of course, monoclonal antibodies are also utilized to clear pathogens from the body also. This is a normal technique. Now, with this I would like to end my lecture. Thank you very much. Hope you have understood what monoclonal antibodies are, how they are very important and have a tremendous use in producing in therapeutic, in diagnostic and various other purposes. Antibodies as such are extremely important because they are a very important part of the immune system. They are the actual reasons which can bind to the antigens, combat the antigenic attack and hence protect our body. They provide, as they say, the kiss of death. Thank you very much.